Face center, let me repeat. Face centered body diagonal is a cube standing on its corner. You see this is a cube standing on its corner? Don't build it off this, build it off the template. This is the face center cube.
We choose one, we choose two.
Yeah.
Who knows? They all look the same.
The colorless is 1, 2, 3. So all the fractions are green. Oh, fractions are green.
How do you know how many balls are there?
I guess you're just following this number.
1 means layer 1. They should all be the same color.
Should we the same color?
Yeah.
Well, those two are different colors.
No.
In the size, in terms of size, right?
Ignore this. I don't know why they made these different colors. They're all the same color. They're all the same atom.
Choose the same size.
Yeah. Don't lose any balls. It's built to scale, so just put it. Yeah.
Oh.
No, it is.
Actual size.
I don't even know.
The spacers shown on the template are the actual size, so just size it up.
Wait, I'm not sure what I'm doing anymore. How do we?
What do you see on the template?
Well, in the template they just give me numbers, but I don't know like.
The numbers tell you what to put. So
how
number
many to
0.
put, right?
Number 0, put a spacer.
I put a spacer.
Yes.
This, the short one or the small one? Or?
The spacer that lines up with the same size as shown on the template. Do you see the spacer on the template? Bottom left.
What's that?
Right there.
Oh,
Where your finger is. Yeah.
But how do we know? Like I know here I have to. do
Whatever
balls.
rod has a 0 on it, put a spacer there. Now your rods look off.
Wait, I'm doing it by the. But how do I know? Like I know this one.
1.
How do I know when to put the?
No,
Because if I
nothing
put
goes there. Now you put spacer, and then you put 2. Everywhere it says a 2, put a 2. Ball there. Okay, this is how you build the body diagonal. Where it says 0, put a spacer. Where it says 1, put a ball.
Which is 2?
Yeah.
Which is 2?
Where it says 2, you first you have to build layer 0, 1 first.
That's not.
And then wherever it says 2, put a ball.
So put all 1 first, and then put 2 in order.
Yes.
It's this one.
But don't forget the spacers.
It's to the actual. Does actually the actual.
Does the color matter?
It's actual size. The color does not matter.
There is no actual size like this.
Well, how about just one person build it and then we'll just show it. You got it done, Keith? All right.
No. Face center, or one of you has to do face center, and another one has to do face center body.
All right, Keith figured it out. What you have is you have 1 ball in the center and 3 spacers. Do you see that? And then you have a triangular layer of balls coming up next, which we call layer 2, and then layer 3, and then it's a repeat of layer 1. You see this is face-centered cubic standing on its corner. Now, let me talk about this for a second.
Versus 0 over 2, what is it?
0 means put a spacer. We need 3 spacers. The spacers are, the size of the spacer is on the page. So all you need is 3 plastic spacers. Where it says 0, put it on that rod. And you need 1 ball.
Capacity, is it the right structure for this one?
It's not complete.
How do you
You didn't finish building it. Just add more layers.
Oh, I just did this one. You're supposed to finish it all. You finish it all. That's it? Yeah? This is 1, 1, in center 1, 1, 1, 2, 2, 2, 2, 2, 2, 2, 2. How do you finish this?
You know what? My calendar. Yes.
On top? How did you know that? How did you know that?
So there's.
Did everyone accomplish building this one?
We're working on it.
Yes?
I'm close.
We're still building. Do you have this one first?
So 0 here.
You have this one.
And another 0.
This one is the, looks like a square. This looks like a square.
Yeah, but then you have to.
So where it says 3.
You have to,
Not 3 balls. That's the 3rd layer of balls.
you have to angle like this.
1 is the 1st layer of balls. 2 is the 2nd layer of balls. All the balls are the same. 3 is the 3rd layer of balls. Well, I got the easy way to build the other one. So just build the first structure, and then we're going to stop. All right. When you build this first structure, do this for me. Lift off the center ball, in the top center ball here. Lift it off and look inside. When you lift off the center ball and look inside, you'll see a little pocket in there. That little pocket's called an octahedral hole because it's surrounded by 6 atoms. The 6 atoms are these red atoms. In the center of these 6 red atoms is a octahedral hole. It's quite small, right? It turns out in the gaps here, do you see the gaps here? On the edges? The gaps on the edges are called octahedral holes also. If you lift off a corner atom, and look at the little pocket that's formed there. The little pocket that's formed there is called a tetrahedral hole. There are 8 of them because there are 8 corners. Underneath each corner atom is a tetrahedral hole. You don't have to memorize this. This is what I was trying to get you to build earlier. Face-centered cubic is the most efficient way of packing spheres. It's also known as cubic closest packing. And so when I tried to get you to build the body diagonal, it was trying to show these layers, the cannonball layers here, like this. This would be an example of face-centered cubic closest packing. That has a high packing efficiency. The void spaces are very small.
There is no this.
There's also another closest packing arrangement. This other closest packing arrangement is called hexagonal closest packing.
First layer.
So, cubic closest packing is also known as face-centered cubic, and we have these. Each layer is what's called a closest pack layer. A closest pack layer is one ball surrounded by as many balls as possible in the layer. If you look at this one ball here, it's surrounded by 2, 4, 6 other balls. Do you see that? There's 6 balls in the plane, and then if you look underneath, there are 3 balls in contact with this, and above this, there are 3 balls. And so we have something called the coordination number. The coordination number of closest packing is 12. So there are 12 bonds here, but they're metallic bonds. And so you don't have to worry about hybrid orbitals, that kind of stuff. And so the difference in packing is the layer sequence, A, B, C, A. And so A, B, C, A is cubic, whereas A, B, A is hexagonal. And the only difference is this triangle above and the triangle below, the 3 balls. If the 3 balls overlap above and below, you know, the next layer I have a choice. I could overlap the bottom layer with the top layer, put a ball here, put a ball here, put a ball here. That's hexagonal. Or I could offset it. Do you see this hole? I could put a ball at the hole, a ball at the hole, a ball at the hole. That's cubic. And so I have choices. I could put, do you see the gap here? The gap is superimposed with the lower layer, or the gap here is not superimposed with the lower layer. I have 3 holes not superimposed and 3 holes superimposed. That gives me the different stacking patterns here. So do you see, all they did was they took this one, the first model you built, and then have you built it standing on the edge. The reason they had you build it standing on the edge is so you could see the layer structure. Layer 1, layer 2, layer 3, layer 1. It's an A, B, C stacking pattern of cubic closest layers. This one looks like a cube with atoms in the face, and so therefore it's called face-center cubic. This is called cubic closest packing. Cubic closest packing and face-center cubic are the same thing. This is a hexagonal closest packing. This is the hexagonal unit cell, which we aren't going to worry about. These are the stacking patterns. HCP for hexagonal closest packing, CCP for cubic closest packing. This is showing how we slice and dice atoms to make them fit into a cube. Internal atom is not sliced and diced. A face atom is sliced in half, an edge atom is sliced into a quarter, and a corner atom is sliced into an eighth wedge. This is showing how we pack the atoms into that cube. This is showing how we determine the crystal structure. We shine X-rays. From how the X-rays are diffracted, we can determine what the structure of the crystal is. Lower order. It's called X-ray diffraction. This is something called Bragg's law. Bragg's law says that there's going to be an angle where we're going to get constructive interference. All the other angles are destructive. And so we need these to be at this angle so that the peaks coincide with the peaks and the valleys coincide with the valleys. If the angle is a little bit different, then no X-rays are diffracted. More complicated structures can be built from simple structures by looking at the holes. The 2 most important holes are the tetrahedral hole and the octahedral hole. If we put smaller atoms in there, we can get things like this. If you look at sodium chloride is a face-centered cubic lattice of chloride ions with sodium ions in all the octahedral holes on the edges and one right in the center. Sodium chloride. Other zinc sulfide has ions in the tetrahedral hole. Calcium fluoride has ions in the tetrahedral holes. Let me show carbon. Take a look at diamond. If you look at diamond, look at the gold atoms here. All these atoms are the same, but if you look at the gold atoms, what lattice does diamond build?
What lattice?
The diamond lattice, if you look at just the gold atoms, can you see it? The gold atoms, you have a gold atom on the corner and one in the middle of the face. This is face-centered cubic. So diamond is described as face-centered cubic with carbon atoms in the tetrahedral holes. That's one way of thinking about diamond. And so a lot of these more complex structures are just built from simpler structures, like that. So there's one last thing. Where did it go? One last thing is called the Born-Haber cycle. You remember I talked about bond energies for ionic lattices? It's made up of a lattice of bonds. And that can actually be measured using this cycle, thermodynamic. And so Hess's law is a way to get lattice energies from data that you already know. The data that you already know would be ionization energy, electron affinities, etc. And from this we can calculate the lattice energy. And so Hess's law application.
Lattice energy is not the same as the bond.
Yes, lattice energy is not the same as bond energies. That's correct. All right, so basically we just have a couple of things left. One, you have a choice. I was going to show this video on how crystal structures are determined, or do you want this video, molecular structure and health? Molecular structure and health? This one I was going to show earlier, but I didn't have the time. Since I have a little bit of time, I'll show it. What's that?
What's the difference between?
One shows you how you use X-rays to figure out the structure of crystals. The other shows you the three-dimensional structure of molecules and their impact on health.
I want to see that.
You want to see that one? No, no safety video. Yeah, just put it.
Wait, should we talk about?
Yeah, we do. Yeah, we don't have enough time.
Oh my god, that looks so... How old is that video? 7 years. Is that a historical piece?
It's old, but good. Yeah.
Did you watch this video when you were in school? Oh!
No, I think this came.
This is a production of the Chemical Education Material Study.
Yeah, that's definitely.
systems is remarkable.
Wow.
More than 1 billion species currently exist. We now know that a principal factor in this large number is the billions of ways that 4 rather simple chemicals can vary their sequence in forming DNA genes. We are also discovering that many illnesses can be identified at the molecular level. This provides information for the design and synthesis of molecules, which can be used to treat the disease. If we examine living cells under the microscope, like these plant cells, or these bacteria, or this animal tissue, we can see the movement, growth, and cell reproduction, which are the result of successions of chemical reactions. In attempting to control these reactions, physicians have used various substances, some from nature, and some synthesized in the laboratory. Many of the compounds found to be useful were discovered by trial and error. One such compound was sulfanilamide. Let's demonstrate the effect of sulfanilamide on bacterial growth. A nutrient medium in a petri dish is used to sustain growth of bacterial organisms. These bacteria, harmful to humans, are carefully transferred from a stock preparation. They are gathered in a wire loop and streaked onto the nutrient medium in both compartments of this dish. A small piece of sterilized filter paper is wet with a sulfanilamide solution and placed on the bacteria in one compartment of the petri dish. The dish is placed in an incubator for 24 hours. During that period, seen here in time-lapse photography, the bacteria multiply and become visible as large colonies everywhere except in the area around the paper disc. The clear zone shows that bacterial growth has been stopped. This inhibition is due to the sulfanilamide from the disc. How does sulfanilamide stop the growth of these harmful bacteria? First, let's examine the function of one of the key molecules that exists in the bacterial cells. It is called para-aminobenzoic acid. Using a geometric symbol retaining the general contours of the molecule, we'll follow its action within the cell. In the cell with the acid is an essential bacterial enzyme. In order for the enzyme to perform its active role, it must first combine with the para-aminobenzoic acid. Only then can the enzyme catalyze the further reactions necessary to cell life. When we examine the structure of the acid molecule and compare it to a molecule of the drug sulfanilamide, we find that they are much alike. This large portion of each is identical. The electron charge distribution in each molecule is also similar, and their general configurations are the same. Using geometrical representations again, let's include sulfanilamide in the bacterial cell along with the normally present acid molecule. The sulfanilamide too can react with the enzyme. But the sulfanilamide not only fails to activate the enzyme as a catalyst, it also prevents the acid from doing so. Thus, metabolism stops, and the bacterial cell dies. The effect of sulfanilamide on cell growth was found by trial and error. But the discovery that sulfanilamide is structurally similar to an essential molecule accounted for the action of the drug. This principle of structural similarity helps us to predict which drugs might be effective against specific diseases. Consider, for example, the use of drugs in the treatment of cancer. By means of tissue culture technique, human cells are kept alive for microscopic observation. Here are normal human skin cells under the microscope. In time-lapse photography, cellular activity appears accelerated. We know of very few differences between the chemistry of normal cells and these cancer cells. One remarkable difference, however, is that the cancer cells grow faster. And another difference is that during the faster growth, the cancer cells use abnormally large amounts of a substance known as uracil. Can a structural modification of the uracil molecule be used to disrupt the fast growth of the cancer tissue? Let's follow some of the steps a biochemist takes in answering such a question. He is familiar with the visible parts of a single cell, like this one undergoing division. We see a set of dark ribbons in the middle of the cell. These dark objects are chromosomes, and they are intimately connected with the process of cell growth. The biochemist has learned to collect chromosomal material from cells, and has found that it is composed of deoxyribonucleic acid, or DNA. Because DNA is a large, complex molecule, it is helpful to break it into its component parts and identify them. These component parts can be separated from each other by the technique of paper chromatography. After treatment with an acid, a small drop of the chromosomal material is placed on this large piece of filter paper.
We're going to check out afterward.
The paper is rolled into a convenient shape for the next procedure. We have selected a solution in which the chromosomal material is readily soluble. This is placed at the bottom of a glass cylinder.
We're going to check out after this video, so if you don't want to watch this video, it's optional. You can come back in about
The solvent
10 minutes.
is drawn up the filter paper by capillary action. After 12 hours, the solvent front has risen this far. The filter paper is removed and allowed to dry. It is then rolled in the opposite direction and put back in a different solvent. This will ensure a further and better separation of the molecular components. These components are not visible under ordinary light, but if we place an ultraviolet light under the dried paper, we can see that the solvent has distributed the components of the chromosomal material. This distribution has occurred because the solvent front competes with adhesion to the paper for the chromosomal materials, so the components move according to their molecular size and solubility. Thus, different compounds derived from the chromosomes are separated. We'll cut out one of these marked areas and dissolve the material from the paper with another solvent. Now we can analyze the dissolved material with an infrared spectrometer. A characteristic spectrum is produced by each component separated in the chromatography procedure. The absorption spectrum produced by one of these extracts is compared with the known absorption spectrum of the chemical thymine. Apparently, chromosomes contain thymine. Similar analysis of the other components shows that each has its own characteristic spectrum. Identification shows that besides thymine, chromosomes contain adenine, guanine, and cytosine. How do our uracil molecules, which are present in such large amounts in cancer cells, relate to the constituents we have extracted from the chromosomes? We know that these constituents, along with other building blocks, are necessary to make DNA molecules, which in turn have a part in forming the chromosomes. Biochemists have shown that uracil is the substance from which thymine is formed. Without uracil, thymine will not form, preventing the synthesis of DNA, which in turn prevents the formation of the chromosome. When we examine the molecular structures of uracil and thymine, we find that there is but one position in which these structures differ. To convert uracil into thymine, a hydrogen atom here must be replaced by a methyl group such as this. Let's symbolize uracil with its hydrogen atom like this to follow its action within a cell. First, a chemical group called a sugar phosphate residue becomes attached to the uracil molecule. The modified uracil compound then forms a complex with an enzyme. Somehow the enzyme converts the hydrogen into a methyl group to form the thymine compound. This molecule is then freed for eventual synthesis into the DNA chain. How can we disrupt this mechanism and stop cell growth? Clearly, one point of attack is the original uracil molecule. This molecule can be altered in many ways. One of the most promising has been to replace this hydrogen with a fluorine to give 5-fluorouracil. 5-fluorouracil has a number of advantages. First, the hydrogen and fluorine are almost the same size, so that both molecules should fit into the enzyme. Second, the chemistry of a carbon-fluorine bond differs from that of a carbon-hydrogen bond, so that the enzyme might find it difficult to replace the fluorine with a methyl group. And finally, the changed polarity may allow the fluorine compound to adhere more firmly to the enzyme than would a uracil compound. Thus, we predict that the 5-fluorouracil, just like the uracil, will attach itself to a sugar phosphate residue and then fit into the same place on the enzyme the uracil would have used. But once in place, the strong bond to fluorine will prevent the enzyme from replacing the fluorine with a methyl group. And finally, we predict that the strong polarity of the 5-fluorouracil will cause such strong adherence to the enzyme that the normal uracil compound will be prevented from reacting, and that, with the enzyme blocked, the essential chromosomal material cannot be made and the cell will die. Let's test this prediction in the laboratory. Here are two cultures of cancer cells. Those on the left have been treated with 5-fluorouracil. Compare the action of these cells to that of the untreated cells on the right. The untreated cells continue to show rapid growth. The 5-fluorouracil treatment is seen to stop cell division, produce cellular enlargement, and eventually contraction and death. But are we sure that the cancer growth is stopped in the manner we predicted? Instead of the 5-fluorouracil blocking the enzyme as we have suggested, and using this means to kill the cell, is it possible that the 5-fluorouracil is incorporated into the DNA, inhibiting its function? To determine where the 5-fluorouracil is acting, in the enzyme or the DNA, let's conduct an experiment on some human cancer cells grown in the laboratory. A A sterile pipette is used to transfer the cells into a large flask which contains a nutrient medium. This permits continued growth so that we will have enough cells for our experiment. The cells are kept in suspension by a magnetic stirrer and incubated for several days. Here is specially prepared 5-fluorouracil containing radioactive carbon-14. By tagging the 5-fluorouracil with these radioactive atoms, we can determine where the 5-fluorouracil goes in a human cell. We'll add a small amount of the radioactive 5-fluorouracil to the living culture. In about half an hour, these radioactive molecules will be incorporated into the cancer cells. The cancer cell suspension is poured into large cups to be spun in a centrifuge. The spinning collects the suspended cancer cells into a pellet on the bottom of the cup. The liquid is discarded. By this means, the cells from large volumes of the culture medium are finally concentrated into one cup. The cells are then resuspended in cold sodium chloride solution to dissolve the DNA. The low temperature retards enzyme action. This cell suspension is placed in a blender. The violent action of the blender fragments the cells to facilitate dissolving DNA. After storage overnight in a refrigerator, the DNA dissolves, and then the solution will be spun very rapidly in a centrifuge. This spinning action separates the residual cell proteins which contain the enzymes. A high-speed centrifuge is required because the large size of the DNA has made the solution viscous. After the centrifuging, the liquid fraction contains the soluble DNA. The pellet contains the undissolved residue. According to our theory, if the 5-fluorouracil is incorporated into the DNA molecule, radioactivity should be detected in the liquid. Otherwise, it should be found in the pellet. The next step is to concentrate the DNA. The liquid is drawn off and injected into alcohol, which precipitates the DNA. These threads of DNA material can be readily collected on a glass stirring rod. sample of the DNA fraction is removed for measurement of its radioactive content. The protein pellet is retrieved from the centrifuge tube. The meter shows that there is no radioactivity in the chromosomal material. However, the protein fraction does show radioactivity. The experiment proves that the radioactive 5-fluorouracil is not incorporated into the DNA. Though the chemistry is oversimplified as portrayed here, it does show that the structural similarity of 5-fluorouracil to uracil is of crucial importance in its ultimate effect, the enzymatic synthesis of genetic material. 5-fluorouracil is currently one of the most effective chemotherapeutic agents for treating certain types of skin cancer and colon cancer. About 40 anti-cancer agents are in clinical use today. For example, chemotherapy with methotrexate can lead to complete remission for 97% of children treated for acute lymphocytic leukemia. But we need even more effective and less toxic chemicals, especially for lung cancers and brain tumors. In the past, most medicines have been discovered by trial and error, even by accident. Aspirin, the most widely used medicine in the world, was used for almost 100 years with no idea as to how it reacted in the body. Modern techniques are allowing us to learn about living systems at the molecular level and what changes cause loss of health. Now some mechanisms are understood. We are beginning to be able to design molecules the living system needs for recovery.
All right, so actually just wanted to talk briefly about the report. You know, if you're going to write a report about this, you want to detail this. You don't want to say, well, I just added some alcohol here. You want to say, well, the alcohol did what, you know, and that kind of stuff. And so that's part of the discussion. In the report, you should discuss what you're doing in the particular steps, what's happening chemically, this type of stuff, you know, rather than just, well, I precipitated out some barium sulfate, this is my percentage, right? And so we want some of the details. So think about that, or keep that in mind when you're thinking about your report. All right, the last thing we're going to do is return the equipment and check out.